And now we have Josh Evans, who, while doing his undergraduate degree at Yale, was part of the Yale Sustainable Food Project. He then went to Copenhagen for 4 years, where he worked in the Nordic Film Lab, Food Lab, established by the owner of Noma. And he's got a book on edible insects, which I'm told is in the book area. And right now he's starting a doctorate in the geography department here at Oxford on the domestication of microbes in fermented food systems. Okay, Josh.
Most of the talks at the symposium this year have been fascinating to me, and most of them, rightly so, have focused on sort of at the human scale of what we tend to think of as landscapes. And so what I'd like to do is try and maybe explore what it could mean for landscapes in general if they existed for non-humans at slightly different scales. And I'm going to focus on microbes in this talk because I think that they lend themselves particularly well to this kind of inquiry. I'd like to try and touch on 3 questions. Certainly I don't hope to answer any of them conclusively, but at least start to explore some of the questions that emerge when we try and think about landscapes beyond the human or more than the human. The first one is somewhat of a simple question, or at least it seems simple, and it's what might landscape mean for non-humans, or particularly microbes? The second one is how can we know these landscapes, given that we ourselves at least are not fully microorganisms, though there's a lot of interesting research emerging that shows that they do form a large part of our bodies and, in some cases, our minds. And the third question is, and I think this is the one that interests me most in some way, is what do we learn about landscapes in general when we explore landscapes at the microbial level? And for those of you who were at Bronwyn and Francis's talk yesterday, I think you'll see that there's a lot of parallels there. I think thanks, I guess, sort of for setting up in advance this idea that, or painting this picture of bacteria as primary agents in making landscapes. This idea that, in many cases, humans aren't the only or even the primary agents that are making the landscapes that we inhabit. So we'll start with the first one. What might landscape mean for microbes? And instead of trying to answer this question directly, I'd like to maybe go back to some of the research that I was doing at Nordic Food Lab in Copenhagen to try and illustrate what this might mean or what this might look like. This is one of my favorite photographs from the work there because I think it very visually illustrates the whole purpose of the space. It was set up as a nonprofit to do open source research in food diversity and quote-unquote deliciousness. It was set up by the chef of Noma, René Redzepi, in 2009. And the sort of overall goal was to explore the gastronomic potential of the Nordic region. And sometimes what this looked like was exploring different raw materials, some of which may have been used, thanks, that doesn't matter, yeah. Some of which may have been used in the region before, but had been forgotten about for various reasons, like wild plants, for example, and trying to resuscitate that knowledge. Other cases of sort of investigating the flavor potential of raw materials involved looking at raw materials that may be used in other parts of the world, in other cultures, but for whatever set of reasons didn't have a very robust tradition of being used in the Nordic region. For example, seaweeds and algae, which of course in many cultures in East Asia are really sort of fundamental ingredients, but in the Nordic region, even though there are many different varieties of algae, they haven't been sort of had such a robust tradition built up around them so far. And so this approach was very much what governed how we were looking at fermentation, trying to look at what kinds of microbes were available and also what kinds of new flavors they could unlock in some of these raw materials, certain heritage varieties of grains and vegetables and meats and shellfish and things like this. I really want to focus in particular on one microbe. The Japanese name is koji. The Latin name is Aspergillus oryzae. If you're It's a very interesting microbe, and it's being used a lot by chefs right now, largely because it produces lots of enzymes, and these enzymes can be used, and are used traditionally, to break down all sorts of different things. So koji is sort of the building block in Japan of producing sake, because the enzymes can break the starches into sugars, so they can be fermented into alcohol. It's the sort of the root of miso and soy sauce because it can break down proteins into amino acids, which give umami taste. It's a really versatile microbe. And if you're interested in more, I recommend that you listen to Nicola's podcast all about koji, which actually just came out, I think, a couple months ago. And they go much more into the science and history of it there. But what I'd like to talk about is what sort of things you can make with it. So this is a photo of 12 different kind of randomly thrown together fermentation trials. All of these are geared towards becoming a kind of soy sauce. And so I put soy in quotes because there isn't actually any soybeans in any of these. But they're sort of made on the template of a soy sauce. So they're using the koji, they're using certain protein-rich legumes, they're using certain starch-rich grains, and also a few other random kind of aromatic things thrown in there for fun, like certain shavings of juniper wood, or dried smoked blueberries, or just to see what happens, because you don't really know what will happen until you try. It should be also said that most of these failed miserably, but that's how experimentation goes in many cases. But one thing that came out of this, which was very exciting to us, was what we ended up calling faux foie, because it was this sauce that sort of rose to the top of this ferment from all of the things breaking down, that had this extremely complex, savory taste that was very reminiscent to us of foie gras, even though, of course, it didn't contain, not only did it contain no foie gras, but it didn't even contain any animal products. It was purely based on those ingredients that you can see there. And if you want to try your hand at making it, the recipe is on the Nordic Food Lab website. But we must move on, because there's a lot of ground to cover. Yes. So I suppose why it's interesting to talk about koji when we try to think about what a landscape would look like for a microbe is because it wants to be in a very different kind of landscape than what is often a comfortable landscape for us, generally speaking. It likes to exist at least 30 degrees Celsius all the time, sometimes higher. It likes to exist at 90 to 100% relative humidity all the time, which if you were to spend 36 to 48 hours fermenting in that kinds of conditions, you probably wouldn't be very comfortable by the end of it. But those are the conditions in which this microbe thrives. So the kind of priorities that it's looking for in the landscape that it's making for itself is very different to the kind of priorities that we have. And this idea of a landscape as a space that is structured by certain priorities is an idea that also came up, I think, when Joshua was speaking, which was a really lovely idea, and I think it kind of ties in here when we're thinking about what that idea might look like also for organisms that aren't human. So moving on to the second question, how we can know microbial landscapes, I want to sort of go through a few examples of microbiologists who are doing really fascinating work to sort of map the landscapes of different kinds of spaces that are very microbially rich or that are involved in food fermentation, to give a sense of what the sort of state of the science is in many regards. And I'll sort of go through three very brief examples. One is a lab that, now Ben has his own lab, but this is another story. Many of you might have heard of Ben Wolf and Rachel Dutton. They're two microbiologists that were working together at Harvard a few years ago, and they did a lot of work in particular on cheese rinds and looking at the different microbial ecologies of different cheese rinds and comparing them to answer all sorts of different questions. We don't really have time to get into them here, and we don't certainly have time to get into all the data that's contained in this chart here, but the only thing I want to sort of talk about putting this up is that you can see there's so many different sort of balances of microbes in different kinds of ecologies. And when we try and understand what sort of factors govern how those balances can change across different times and spaces, then it starts to become really clear that we as humans, when we're making cheese, are not necessarily the ones that are actually making those landscapes all the time. We might be kind of making proposals to the microbes based on the conditions that we put them in, but ultimately it's actually the microbes who are doing the work. And it's the microbes who are making those landscapes change and fluctuate depending on what kinds of things they eat, what kinds of temperature and humidity they thrive in, and what kinds of things they don't really like in their environment. A second really interesting lab that's doing work sort of in this sort of area is David Mills' lab at University of California in Davis. And especially one of his recent PhD students, Nick Bokulich, has done so many fascinating papers, essentially using metagenomic sequencing, which was similar techniques to what Ben and Rachel were doing, except instead of looking at a fermented food product, they were looking at the spaces in which fermentation happens. So they've published papers on breweries and wineries and cheese cellars and sake breweries and all sorts of different spaces in which fermented foods are made, and one of their sort of overall goals, it seems, is to use this set of techniques of metagenomic sequencing, whereby you can take samples from different spaces or organisms and then sequence all of the DNA in that sample and then compare it with a database to try and understand how the DNA groups together into different strains of organisms. And what you see here, this is taken from one of their papers on a beer brewery, where they took samples from all the different parts of the brewery, and the sort of heat map that you get, the more intense the color is, the more diverse and the more, I think this chart, was diversity. They also have charts for cell count. So you can see the spaces that have more intense color are spaces where there were more microbes coming into the space, on the left from hops that were coming into the brewery, and on the right from the grain that was coming into the brewery in the form of malt. And so this is still in some way very rudimentary, but what we start to see from these maps is that not all microbes exist everywhere in the same degrees. And there are reasons why they will gather in certain parts of the spaces in which they're fermenting. Now, a third one that's being done here at Oxford, actually, it's a project called the Interdisciplinary Microbiome project. And essentially, they're using this very similar techniques, but they're giving them into the hands of normal people to take home and say, what do you want to know about your domestic spaces? What kinds of questions would you ask to your own kind of domestic microbiome? And so it's a similar kind of idea of a heat map here. And unfortunately, I don't have the other slide to compare it to because it's not published yet. But what's really interesting here is that the spaces in the kitchen that most sort of lay people thought would be most microbially diverse were not the spaces necessarily that actually ended up being most microbially diverse. So these techniques can sort of give us new insights into what kinds of landscapes exist for microbes rather than what we imagine might exist for microbes. Yes, thank you. Perfect. So when we start to really get into the nitty-gritty of how these techniques work, in some way the question splits in two. Because on one hand, we have a question of what the landscapes of microbes are like. These would be, many of the questions that the Mills Lab and Ben and Rachel and Jamie Lorimer at the Interdisciplinary Microbiome Project have been working on. But actually, that's a slightly different question to what it would sort of feel like to be a microbe in your own landscape, which is kind of a question that sort of practicing fermentation addresses slightly more directly. And so what we can see is that by integrating these different techniques, using metagenomics to understand what microbes are out there, or in different kind of shared spaces, and also using fermentation as a set of practices to listen and respond and collaborate with microorganisms, we start to get a more rich picture in some way of what it would mean to sort of have landscapes happening for different kinds of organisms, more than just humans. Now, in the last few minutes, I want to touch on some of the more general themes, I think, that emerge from this kind of inquiry by integrating more traditional knowledge of fermentation practice, culinary knowledge of using it in kitchens, and some of the scientific knowledge that's being developed by using metagenomics to address what might we learn about landscapes in general when we explore landscapes at the microbial level. One thing that emerges very clearly, I think, is that landscapes can emerge at different scales, both different scales of space, thinking about the landscape of a cheese rind. For us, it's not quite the same spatial scale as the landscape of, olive groves or vineyards in the Mediterranean. But it's a kind of landscape nonetheless, because there are certain biotic and abiotic forces that are shaping what it looks like and what it tastes like. Another thing that emerges from this kind of inquiry is that landscapes change. Now, this might seem like a kind of banal thing to say, because many of the papers in the symposium have also talked about this idea. But I think what microbes allow us to see very clearly, or particularly clearly, is how landscapes change, because in many cases, their landscapes are changing very rapidly. So if we look at a lot of kinds of fermentations, either the cheese rinds or sake, for example, what you see is not only multiple kinds of microbial communities vying for the same resources, but they're also succeeding each other over time, which was sort of that graph that I didn't really touch on that happened earlier briefly, where yeast will grow and then it will reach a certain alcohol level and then it will die off, and then it will allow certain bacteria to come in later and then they'll die off. And so the landscape of a sort of fermented food system can change even over the course of a few days. And that idea, I think, extends more broadly to landscapes at the human scale, and in some way it can help us think about change that might happen slightly slower, which of course the stakes of that are very high, because thinking about climate change, for example, the scale of it is so enormous, but if we can kind of break it down and think about it at a lower scale, it might help us make it easier to think about in some way. A third thing, and perhaps a last sort of reflection on what microbial landscapes might teach us about landscapes in general, is that different landscapes organize space differently. Or put another way, they make different kinds of spatial distinctions. So when we think about human landscapes, it's very tempting, I think, a lot of the time to fall into using very, perhaps not very rigorous analytical distinctions like natural and artificial, or built and natural, or found and made, or inside and outside. These kinds of distinctions that, at our level of experience, seem to make a lot of intuitive sense, but don't necessarily hold up to deeper kind of analytical scrutiny. So an example of this might be something like, a kitchen for us. I think few people would dispute that a kitchen is a kind of made space. But what some of the research in the microbiome of domestic spaces tells us is that for microbes, it's actually not a made space, it's a found space, because they're just sort of flourishing there based on conditions that we make for them. Whereas if we flip that around and think about, for example, the forest floor of a primeval old-growth forest, few humans would say that that is a made landscape in the human sense, but in terms of microbes, they're the ones that are actually making that humus. They are the ones, the bacteria and the fungi, that are making that forest floor a landscape in the first place. Yes, thank you. I think this might be even more clear as a distinction between the inside and the outside. If we think about the difference between the vineyard and the winery, for example, at our level, when we think about what that landscape looks like for humans, the distinction between the outside, the vineyard, and the inside, the winery, is very clear. But if you're thinking of it from the microbes' point of view, there's so much exchange happening between inside and outside. Yeasts are coming from the winery out into the vineyard, they're coming from the vineyard into the winery, and so that distinction might actually not hold up in the same way at different scales of space and time. So to conclude, I suppose this has been less a foolproof set of arguments and more, I think, a set of reflections that we can think about when we look at what happens in the interesting space between science and cooking, that microbial landscapes of fermented food systems in particular can maybe in some ways make us think about landscapes in general differently. And I think that can be a really kind of fertile exercise. So thanks, and if you have any sort of questions or you think this was all wrong, please let me know. I'd like to hear.
Great. Okay,